Assalamualaikum. My name is Muhammad Izaz Nazim bin J.Henipa. So, today I will present about the pollination and fertilization process and the life cycle. So now we add the second slide, which is what is pollination. So, pollination is defined as the transfer of pollen grains from the anther, which is the male part of the flower, to the stigma, the female part of the flower. This might seem like a simple process, but it's actually the first and most critical step in sexual reproduction of flowering plant. Here are the key points to remember. So, we start from the first one. Pollination is the very first step in sexual reproduction. Without it, plant cannot reproduce sexually. Secondly, pollen grains contain the male gametes, which are essentially the plant's sperm cell. Thirdly, this process is essential for plant reproduction and seed formation. And finally, pollination can occur within the same flower or between different flowers. So, why is pollination so important? Well, without pollination, plants cannot reproduce sexually, produce seeds, or maintain genetic diversity. Next is slide number 3. So, in these slides, we will talk about types of pollinations. There are two main types of pollination: self-pollination and cross-pollination, and each has its own advantages and characteristic. So, we start from the self-pollination. Self-pollination occurs when pollen is transferred within the same flower. This is called autogamy, or between different flower on the same plant, which is called geotonogamy. The main advantage of self-pollination is that it ensures reproduction even when pollinators are not available. However, it results in less genetic variation because the offspring are genetically similar to the parent plant. Cross-pollination, on the other hand, involve pollen transfer between flowers of different plants of the same species. This type requires external agent, which we call pollinators, to move the pollen from the plant to another. The major advantage of cross-pollination is that it promotes genetic diversity, which often produce stronger, more resilient offsprings that can better adapt to environment or change. Okay, so we move to the next slide. Slide 4, about the pollinators. So, pollinators are the external agent that help transfer pollen from anther to stigma, making cross-pollination possible. There are several types of pollinators, each adapted to different plant species. So we will discuss about different types. The most common pollinators are insects, including bees, butterflies, moths, and beetles. Bees are particularly important. They are responsible for pollinating about 80% of flowering plant. We also have wind pollination, called anemophily, which is common in grasses, trees, and cereals like wheat and corn. Water pollination, or hydrophily, occurs in aquatic plants where pollen is carried by water currents. And finally, we have animal pollinators like birds, bats, and small mammals. This is called zoophily. So, the importance of pollinators cannot be overstated. About 80% of flowering plant depend on animal pollinators for reproduction. Okay, next we move to the next slide, which is the pollen grain development. The development of pollen grains is called microsporogenesis, and it's a fascinating process that occurs in the anthers of flowers. The process begins with diploid cells called microsporocyte. In this enters this cell undergo meiosis, which is type of cell division that reduces chromosome number by half. This meiosis produces 4 haploid microspores from each microcyte. These microspores when developed into mature pollen grains. A mature pollen grain has a very specific structure. It contains two important cells, two generative cells, which is the two sperm and tube cell which grow into pollen tube, essential for fertilization. Okay, next we talk about female gametophyte development. The development of female gametophyte is called megasporogenesis, and it occurs within the ovules of the flower. So, I will explain the process. Okay, so the process starts with a diploid cell called a megasporocyte located inside the ovule. Like in pollen development, this cell undergoes meiosis to produce 4 haploid megaspores. However, typically only one of these megaspores survive and develop further. These surviving megaspores divide multiple times to form the mature embryo sac. So, the mature embryo sac contains 8 nuclei with very specific function. The most important is one egg cell, which serves as the female gamete. So, there are also 2 polar nuclei, which will play a crucial role in forming the endosperm, the food storage tissue. The remaining 5 nuclei form support structure that help with the fertilization process. Okay, next we will talk about the double fertilization. The double fertilization is one of the most distinctive features of the flowering plant, and it's called double because it involves 2 separate fertilization events occurring almost simultaneously. So, when a pollen grain lands on a compatible stigma, it begins to germinate. The tube cell grows into a pollen tube that penetrates through the style and grows toward the ovary. Meanwhile, the generative cells divide to form 2 sperm cells. The pollen tube eventually reach the embryo sac and release the sperm cells. Here's where it gets interesting. 2 fertilization occurs. In the first fertilization, one sperm cell fuses with the egg cell to form a zygote. This zygote is diploid, containing chromosome from both parents, and it will develop into the plant embryo. In the second fertilization, the other sperm cell fuse with the 2 polar nuclei to form the endosperm. This endosperm is triploid. It has 3 sets of chromosome and serves as food storage for the developing embryo. Okay, so next is the plant life cycle. The plant life cycle demonstrates what we call alternation of generations, alternating between diploid and haploid phase. So, the cycle begins with the sporophytes phase, where the mature flowering plant is diploid. This plant produces spores through meiosis in its reproductive organs. Next is the gametophyte phase, where these haploid spores develop into either pollen grains, the male gametophytes, or embryo sac. The female gametodes. Then comes fertilization, where the male and female gametes fuse to restore the diploid condition. So, we move to the last slide, which is the summary and the conclusion. Let's recap the key takeaways from our discussion today. So, first, pollination is the transfer of pollen from anther to stigma, and it's the critical first step in plant reproduction. Second, we learn that self-pollination and cross-pollination each have distinct advantages. Self-pollination ensures reproduction while cross-pollination promotes genetic diversity. Thirdly, pollinators are essential for many plant species, with bees being particularly important for food crop production. Next, double fertilization is unique feature of flowering plant that efficiently produce both an embryo and its food supply. Finally, the plant life cycle alternates between diploid and haploid phase, with the sporophyte being dominant.